And so that actually ties in really nicely to my next question here around where, how much we've learned. You've already talked about how we've gotten here to date, and some of the important elements of why LNP and mRNA work really nicely together. They're scalable. We can understand their potency, right? Cationic lipid has been very helpful. But when it comes to looking at some of the products that have come before, right? We have the mRNA vaccines and we have Onpattro. Can you guys tell me a little bit more about what big learnings, that you took away from those that have been particularly instructive to the work that you're doing today on your own therapeutics and your own vaccines? Russell, we'll start with you on the vaccine side. I mean, are there takeaways that you can have even from the Onpattro side, right? The therapeutic side and the work that you're doing in formulation for vaccines or, from the mRNA COVID vaccines?
Right. Well, it's just to put it straightforward. I mean, right now it seems like this great and fairly cohesive story, right? In late 2010s, right? You had kind of the initiation, really 2005, right? You began to see, DNA, and which is really kind of the grandfather of all of these lipids that became remarkably versatile with siRNA delivery, and then there was a wave of companies that then kind of followed. Kind of right after that, Cerna being acquired by Merck, Roche acquiring mRNA's, and Alnylam collaboration with Tecmira at the time, right? And so it was a field that was moving very quickly. But I think it's also important to keep in mind that there was no contemplation of mRNA therapeutics at that point, right? So CureVac was kind of the first mover in that moment, and so just from a historical perspective, and they were looking at just naked mRNA that was delivered with the protein mRNA that was really used to just adjuvant, right? And the idea was that you could just get barely enough expression to elicit an immune response. And they went to clinic and failed, right? And then they went back, right? And it was kind of this strange story, but the overlap of mRNA and siRNA was not close, right? It was really dispergent kind of endeavors to kind of look at what was happening. So I think what Onpattro really showed you was that actually there was a high degree of interest, or at least a sustainable interest in new technologies and the promise that they could give you over genetic medicines. I think that is maybe a key takeaway. But it was really not in any closer, really related efforts that you saw a crossover to mRNA and LNP applications, right? And there was a lot of reasons for that, right? mRNA is such a different molecule than siRNA that really, it was a big jump getting to that point. But then once you saw a movement over in that direction, and really this was happening in about 2012, right? There was a publication in PNAS from Novartis looking at self-amplifying mRNA, and then you saw pretty, initially gradual, but then very direct applications of lipid nanoparticles with mRNA therapeutics. And that included, efforts by, BioNTech, Moderna, and eventually CureVac, who were kind of the most recognizable. And then you had in the background, which maybe they weren't in the background so much, but initially it was Shire, right? And Rana, and then, and then that kind of evolution into now what is part of Sanofi, right? So there's a lot of kind of history in all of that interactions, and I think it's really pretty cool to kind of see how that development has happened and watch it over the last 10 years. But in any case, so getting back to the main point, I think it was the best thing about Onpattro was that there was enough interest in genetic-based medicines that were non-viral, that there was, it kind of laid a good, your runway, for emerging mRNA technologies to then kind of take off. So I think I'll leave it at that, even though it's a little bit more general. It's not super directed at LNPs, but I'll leave it there.
That's fine. No, I think it's the notion, right? Build it and they will come in some way. It really does lay the framework to have that successful industry, which we really definitely need, right? We need that initial leader, right, to kind of get things kicked off, and the scientific research to build on top of that, right? So that everybody can kind of get, oh, we're being recognized. And I think that recognition can also really help an industry take some roots. So that's a great point. Alok, what about you? You know, you're working in the oncology space in particular, on, you know, working in the innovative, you know, mRNA space. What key takeaways have you learned that are helping you today do the work that you're doing at Strand based off of the mRNA vaccines and Onpattro?
Yeah, so to your point, as you said, we are in the therapeutic space, and so I'll go back to how LNPs evolve, right? And just a brief note on, this was not a straightforward puzzle to solve, right? So, for example, MC3, if it from all the way from DOTAP to MC3 was a 10-year journey, and then from MC3 to, if you take MC3 and try to use for mRNA, it's not going to work, right? You'll need these better lipids. So that journey again was 5 to 7 years. So it takes time to solve the puzzle is one. The other is, when you compare and contrast the vaccine versus therapeutic world, vaccine is low doses versus therapeutic is at least a log-fold higher, the bare minimum. And so that's where the tolerability aspect really kicks in, right? I mean, vaccine also needs to be, all the lipids that you put in need to be biodegradable, cleared out of the body and all of that, right? But in vaccine, it becomes the key driver for a program to become successful. And so that's what we have been focusing on here at Strand, right? Because the more doses, the more tolerated your particle is, the less you have to worry about the tox coming from the product itself, right? Everything has a tox, and you have to be mindful of the tox aspect when you go into the therapeutic side of the spectrum when it comes to LNP. Again, I mean, and one big advantage with non-viral, which a lot of people should appreciate, is that you can redose, right? Which others don't, and that's a key. You can go, for non-responders and stuff like that in oncology. It really becomes important that you can keep going back in until you get a response.
Yeah.
So can I jump in here one second? Because
yes, absolutely.
I think Alok noted like a huge advantage, which is safety, right? Safety. So I think what we saw in the kind of the evolution of LNPs was really that you have to manage two ends to get a successful LNP. And it's true with both vaccines and also therapeutics, really. You need something that has a fairly wide therapeutic window, right? And you drive that with two things, right? Potency, right? Because that ultimately determines success, and safety, and because that really, it gives you the possibility of moving things forward, right? It opens up the possibility to redose it. So I think those two areas become very pivotal in a successful product.